Namaste students, I am Jayoti Dr. Aziz Ahmed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jayoti Vidyapeeth, Women's University, Jaipur. Today I am going to discuss about the topic extreme environment, and this is the continuous part of the extreme environment. So today the first extreme environment which I am going to discuss is thermophiles. Thermophiles is also the extreme environment. So Greek word thermo is a Greek word which means the heat. And the phile is also a Greek word which means love. So thermophiles is the organism love heat. Love heat. Means they are the organisms which love to heat. So come to the definition. A thermophile is an organism that thrives at relatively high temperature. Thrives at high temperature conditions. Rock suggested a definition which is thermophile is an organism that is capable of living at temperature 8 or near maximum for the taxonomic group of which it is a part. The definition of thermophiles. Now talk about the occurrence of the thermophiles. They are found in the compost. They are found in the compost. They are found in sun-heated soils area, and they are found in the terrestrial hot springs area, and they are found in the submarine hypothermal vents. And okay, so these are the things. They are also found in the geothermally heated oil reserve. Also found in the oil wells, okay, where the temperature is high. Various geothermally heated regions of the earth, such as the hot springs, like those in the Yellowstone National Park. The diversity of bacteria of a hot spring in the case is also a form of the thermophilic microorganism. So these are the examples where the thermophilic thermophilic microorganisms are maybe any fungus, any bacteria, any yeast. They can be any kind of microorganisms. So what are the diversity of these thermophilic microorganisms? So if first one is the fungi. Some of the fungi are thermophiles. What are these fungi? For example, the zygomycetes. The zygomycetes have the genus which have rhizomuca and rhizomuca uscillus. So these are the fungi which are thermophilic in nature. Similarly, ascomycetes. In the ascomycetes, Itomian thermophile is a Thermoastus auranticus is also a thermophile. Retiomyces thermophilus, next example, Melanocarbus, Albomyces, and Teluromyces thermophilus, uh, and Telluaria terrestris is also example of the ascomycetes. Next example is the Basidiomycetes. Veniocorti Crassospolium is an example of fungal, and Hypomycetum is also an example, which have the Acremonium, Leptomonsis, and Tribolium thermophilum. Similarly, some more Phosphora thermophylla and Thermomyces leguminosus, and Cytridium thermophyllum and Melinensia cinnamomyia. So these are the some examples of the fungi which are included into the uh, thermophytic. Some algae are also, for example, Chintus exigua, Cetria, and Cynidium, Caladium. So these are the examples of the algae and the protozoa, which are also thermophilic. The Pothuria species, Oxytricha fella, and sulfur methansis, okay, and Tetraanima piriformis is also an example of the protozoa, which is thermophilic in nature, and Cyclidium cytoxus is also example, and Media fowleri is also an example of the thermophilic microorganism. Some bacteria and archibacteria are also the example of the thermophilic microorganism. We'll discuss one more lecture about the archibacteria, and bacteria and archibacteria have been classified based on their optimum temperature requirement. For example, they can also be found in the moderate, extreme, and hyperthermophiles. So, what are the moderate microorganisms? For example, the Bacillus Galvoliticus, uh, Thermoactinomyces vulgaris, Clostridium thermocervicarum, and Thermoaerobacter ethanococcus. So these are the examples of the thermophilic bacteria which lives at moderate high temperature. Some environment which are which have the temperature higher than the moderate thermo- thermos environment are the extreme thermophilic environment. For example, the microbes which include the Thermus aquaticus, Thermus thermophilus, Thermodose sulfibacterium communis, and Sulfolubus acidoalkibulus. So these are the examples. Next example there is a hypothermophilus. Next, hypothermophilus. So bacteria and the archaebacteria which are found in hypothermophilus include the Methanococcus, Gemstag, Acidnonouscus, Inferons, Archaeolobus, Profundus, Methanophilus, Candidai, Pyrobacterium, Isandicus, Infuriaceus, Pyrodictum, Optum. So these are the examples of the hypothermophiles. What are the reasons? What are the conditions by which these thermophiles get they adapt, get adapted to the extreme environment? What are the general features? Have particular some enzymes. They have some enzymes which make them to adapt to these high extreme environment. So what are these conditions? They have these microorganisms have some specific enzymes which make them to survive at high temperature. Actually, these enzymes can be stable. They can be stable at high temperature. And the normal organism like us, we cannot survive at high temperature because we do not have enzymes which can survive high temperature. For any kind of activity, we need some enzymes. Okay? And enzymes perform their activity at particular temperature, at suitable temperature, at suitable temperature. So if they do not want, they do not have these suitable environment, they cannot function properly. But extreme, these microorganisms which are present in the extreme environment have the enzymes which can survive at high temperature. So these are the reasons why they are surviving due to the presence of these kind of enzymes. Okay. They also the second reason is that they have adaptation of protein synthesis at high temperature. They can mm, have the ability to synthesize the protein at high temperature. And what are needed for the protein synthesis? Enzymes are needed, and they have enzymes which can survive high temperature. Next adaptation is of membrane lipid composition. The membrane lipid composition of these microorganisms are different from the normal environment microorganisms. The, the environment microorganisms which live on the action environment have membrane with different lipid composition, okay, which make them to survive at high temperature. Most hypothermophiles are anaerobic and chemolithotropic, which make them adapt at high temperature because they need food. Any microorganism needs some nutrients, but if they do not find any nutrients, what how will they will utilize these nutrients? Okay. So the anaerobic nature of these action these microbes which are present in the action environment make them to utilize more nutrients. Similarly, they are chemolithotropic so that they are not dependent on the some organic molecules. They can utilize anything. One more thing, there are no particular carbon use or energy generation pathway in these organisms, and they are exclusively limited to the growth at high temperature, and they have not particular specific features for carbon and energy generation pathway. They have general mechanism, and they have mechanisms which are linked to the high temperatures. For any microorganism, lipid, nucleic acid, and proteins are generally susceptible to heat. We know that. The most important thing, the lipids, nucleic acids, and proteins are susceptible to the heat. They cannot survive at the high temperature conditions. Therefore, there is no single factor that enables all thermovials to grow at elevated temperatures. But they can still survive. They are adapted to these conditions. The proteins, the nucleic acids, okay, the lipids are adapted to the extreme environment. So that's why that is the reason why they can survive the high temperature. Talk about the membrane of the lipid of the thermophores. They contain more saturated and straight chain fatty acids than the miniphilic microorganisms, which allow the thermophiles to grow at high temperatures by providing the right degree of the fluidity needed to the membrane function. So these features provide fluidity to the membrane so that they can survive at high extreme environment. Hyperthermophiles have a reserve gyrase enzyme. Okay, they have an enzyme which is known as the gyrase enzyme. It is a type 1 topoisomerase enzyme that causes positive supercoiling and therefore may stabilize the DNA. It is a type of topoisomerase enzyme that causes positive supercoiling. Okay, so it stabilizes the DNA. So that is the reason why the DNA can survive. Okay. Similarly, some heat shock protein chichrons are highly present and likely to play a key role in the stabilization and refolding the proteins. So these are the reasons why the proteins are resistant. Here I must share the screen so that you can understand the structure of the membrane structure of the thermophilic organism. So this is the structure of cell membrane of the thermophilic organism. The top layer is of a phospholipid layer of thermophils which have ether linkages, isoprenoid diether and tetraether structures. And similarly, the lower layer of phospholipid layer of eukaryotic organisms like bacteria, they have ester linkages. And the lipid bilayer labeled 9 eukaryotic and bacteria, and the lipid monolayer. So this is the feature by which these organisms can survive at high temperature conditions. So this is all about the thermophiles, which I discussed in this video. Uh, in this video, I discuss about the thermophiles, what are the thermophiles and what are the diversity, like what are the fungal species, which fungal species are included into the thermophiles, which algae are included, which protozoa are included into the thermophiles, and similarly the bacteria and archaea which are included into the thermophiles. Um, I also describe the adaptation of the thermophilic microorganisms, how they are adapted to the extreme environment, extreme high temperature. So, what kind of question which can arise? The question which can arise is that explain the thermophiles, explain the thermophilic microorganisms, and explain them with their diversity. What are the method for the adaptation to the extreme environment? So I hope that students will better understand all these things and they will answer all these things. So my lecture is over here. Next video I will discuss some more extreme environment and the microorganisms. So my lecture is over here. Thank you very much.